Benjamin.
Okay, so thanks, Owen, and thanks everyone for being here. So today I'm going to talk about the chromatin conformation, how to detect it, how to analyze the data you're going to get from the detection of it, and how I used it in the context of the project we're working on. But first, I would like to ask a question. It's a simple one. What makes us human? So if we compare our genome from the genome of a chimp, we see that it differs from about 1 to 3%, but phenotypically, humans and chimps are really different. But if instead of looking at all our genome, we just look at our enhancers, which is a sequence of your DNA that interacts with the promoters of genes to promote their activations, we see that 16% of them differs between human and chimp. And if we express the human-specific enhancers or the chimp-specific enhancers in mice, they produce a gene expression that's typical for humans or for chimp. So the answer to the question is simple. What makes us human? Well, it's the precise spatial and temporal regulation of gene transcription by enhancers. And this is regulated by the spatial organization of your chromatin, which is going to be the topic for today. So let's start by talking about what chromatin organization is and how it's organized in our cells. So we often think of DNA as just a sequence of A, T, C, G, but DNA is physically present in our cells. It's a molecule. It needs to be stored. It needs to be organized for it to function well. So where is our DNA in our cells? Well, it's in the nucleus of our cells. And then within the nucleus, all the chromosomes that form our genome are organized within the chromosome territory, each of the chromosomes having their own space in the nucleus of our cells. Then within this territory, the actively transcribed chromatin is going to segregate itself from the silent chromatin in what we call compartments, A for active and B for inactive. This segregation is thought to happen because of the biophysical properties of chromatin when it's active or inactive. And then within these compartments are found what we call topologically associated domains or TADs, which are segments of your DNA of about 100 kilobase pair to 1 to 2 million base pair that interacts more strongly with them cells than with what's around it. So our TADs form, and what is the function of TADs? Well, TADs are formed by a protein complex called cohesin that is shaped like a ring that is going to handcuff two strands of the chromatin that is again going to go through it to form a loop until the cohesin reaches CTCF that is found on the boundaries of these loops, and that is going to block the extrusion of the chromatin through cohesin. So this is going to result in a domain that is attached by the cohesin and bound to the CTCF at the edge. And within the domains, the interaction between promoter and promoter or be between enhancer and promoter is stronger within it. And the enhancers that are outside of these TADs won't be able to influence the promoters within another TAD. So if you have a signal or a stress conditions that is going to activate an enhancer from another TAD, well, it's going to activate the genes within this TAD, but it's not going to affect the genes in another TAD. So this spatial distribution of the chromatin allows for specific changes in gene expression or epigenetic changes that won't affect other genes. And this is really important for the development of, well, humans and animals, because as our cells differentiate and as we develop, these domains becomes more and more clear and more insulated, allowing for cell type specific interaction between enhancer and promoter to be promoted in these specific cell types and blocked in other cell type as their domains will differ. So if you have an embryo and you knock out all the CDCF in it, it won't be able to properly isolate these domains, so it won't be able to properly differentiate, so the embryo is going to die. So the proper insulation of your domain is critical for cellular identity, but it's also critical for the normal functions of your cells. As if, for example, one of these insulator site is mutated and cannot bind to CDCF, and it happens that it was insulating an enhancer that otherwise could contact an oncogene, was going to promote the expression of this oncogene, and as in this paper, the insulator sites here allow the enhancer to interact with PDGFRA, which promoted cancer progression. So insulator sites are often found mutated in cancer, but what happens if instead of being the site where CDCF is bound that is changed, it's CDCF itself that is changed. Either it's a mutant or if its levels are lower so that it cannot bind every site it's supposed to. Well, CDCF was shown to be a haploinsufficient tumor suppressor gene, as mice with a hemizygous deletion of CDCF have increased rate of spontaneous cancers, and also in humans, CDCF is found in one of the most common single allele deletion found in cancer. It's found in more than half of all breast tumors, and it's associated with cancer progression and metastasis. However, despite everything I showed you before, it is not known the clear function of why its haploinsufficiency promote cancer progression. So in our lab, our models and the project I work on is that CDCF copy number loss will lead to specific patterns of epigenetic reprogramming, which will promote cancer progression and can be targeted therapeutically. So in other words, when the levels of CDCF go low, the weaker binding sites of CDCF in our genome won't be able to bind to CDCF. So the isolation that happens here will be lost in the knockout, which can allow then enhancers to contact to oncogenes, or it can allow a TAD where the chromatin state in it was mostly closed to spread to a TAD where it was open. So either increasing the expression of an oncogene or silencing a tumor suppressor gene. And since only the weaker sites of binding of CDCF will lose the binding to CDCF, in every cell, the same sites are going to be affected, and therefore we can know what are the oncogenes that are promoted this way, and we can find a therapy against it. So we're going to take a little break of TADs and the domains to give a background of my project and talk about cancer and CDCF for a few slides. So first, I used CRISPR-Cas9 to do a hemizygous knockout of CDCF and MCF10A cells, which are epithelial mammary cell lines that are immortalized but not transformed, as wanted to look at the impact of the CDCF hemizygous knockout for early event of cancer progression. And although the knockout was not enough to transform our cells, it was enough to promote some really important oncogenic phenotype, as our knockout cells were a lot more invasive than the wild type cells. And invasiveness for epithelial tumors is really important, as it allows the cells to break through the membrane that surrounds them to promote the cancer to a stage that is higher and more often, deadlier than when the tumor is within the membrane. And also, the knockout of CDCF alters the ability of the cells to grow in 3D, making them grow in bigger, deformed, and filled mammospheres like a cancer cell would do. So then wanted to look at the changes in gene expression that were driving this oncogenicity. So we did a RNA-seq. And when we did a gene set enrichment analysis of our RNA-seq, we saw that the genes involved the PI3K pathway are upregulated in our knockouts compared to our wild-type cells. And the PI3K pathway is a non-oncogenic pathway that promotes cell survival, growth, and also invasiveness. So its upregulation can explain the oncogenic phenotypes we were seeing in our cells. So we decided to inhibit it therapeutically by using taurine-1, which inhibit a kinase that is central to the PI3K pathway, which is mTOR. And we saw that inhibition of mTOR with taurine-1 inhibited its effect, since its downstream effector for ABP was not phosphorylated following the treatment with taurine-1. And the treatment with taurine-1 also reduced the invasiveness of our cells, specifically compared to other cancer cells, such as MDMB231. And another interesting effect of taurine-1 in our model was the reduced expression of SNAIL. So SNAIL is a very potent oncogene, a master regulator of EMT that promotes invasiveness and metastasis in the models of cancer in humans. So it's strongly upregulated in our knockout, and with taurine-1, its expression is reduced. And SNAIL itself, without the inhibition of PI3K pathway, by simply reducing the levels of the SNAIL, we're able to reduce the level of invasiveness in our cells. So taurine-1 not only inhibits a pathway that is driving the oncogenicity of our cells, but also it reduces the level that is upregulated in our cells that also promotes the oncogenicity of our cells. And the next step in our model was to do a ChIP-seq on CTCF to look at the altered sites of binding of CTCF around genes that were either up or down regulated. And we did see loss of CTCF binding in our genome on about 5,000 lost sites. And these lost sites were found around genes involved in the PI3K pathway and on the promoter of SNAIL. So now we have the oncogenic phenotypes driven by the loss of CTCF, the changes in gene expression that drives these phenotypes, and we know that there are altered sites of CTCF binding around these genes. But as of then, we didn't have a clear link between how these lost sites of CTCF leads to changes in gene expression. And that's why we decided to do a IC and to look at chromatin conformation to establish a direct link between the changes in CTCF and the changes in gene expression. So we performed the IC. And in the section here, I'm gonna go in details over what the IC is, how to perform it, how to optimize it, and then how to analyze it. So IC is a technique where you're going to cross-link the DNA in your cells, allowing the DNA that is physically close to one another to be attached. And then you're going to cut this DNA, you're going to mark it, and you're gonna attach it. So the DNA that was physically close to one another is now a continuous sequence. So when you share it with sonication and you pull down the mark, you're gonna end up with fragments that one end of it is gonna be somewhere on your genome, and the other end of it is gonna be somewhere else on the genome that was physically close to the other place. So then you sequence it, you map it, and you know which segment of your genome was close to which other. And before you do a IC, it's important to know that there are many other ways to look at the chromatin conformation, which all differs from the IC. So if you want to look at all your genome, well, you want to do a IC and look at everything. But if you only want to look at loops that are formed by a specific protein, you can do a iChip, where you pull down the protein before, and then you do a experiment that's very similar to IC. Or if you only want to look at a region, you can do a IC that, well, a protocol that's very similar to IC, but before you send your read to sequence, you amplify or select only the regions you're interested in. So with this, you have lesser reads, so you can have a specific reading of only this reagent without having to sequence a lot. So it reduces the cost of your sequencing if you're looking specifically at a region or at a protein. But if you want to do like me and look at everything, then you do the IC. So the first step is to cross-link. So you cross-link with formaldehyde to attach the DNA that is close together, and then you isolate your nuclei if you want to do it in situ. In other protocols, they're going to do it in what we call a dilution ISC, where they don't isolate the nuclei, they simply extract the cross-link DNA and they use the enzymes on it, then cut it and attach it. However, if you do it this way, since all your DNA is floating around, you're going to have a lot of random ligation just because a part of your DNA just happens to be floating by the other part when you attach it. So if you do AIC like this, your background is going to be a lot higher. So now almost everybody do it in situ, so where your DNA is still in the nucleus of your cells. So it's harder for the enzymes to go in, but the nucleus is still going to provide a structure to it that will stop all the random ligation that could happen in the dilution ISC. So when you do that, it's important to make sure that the conditions you're using for lysis are going to be good enough for the enzymes to go in. And if you flash freeze your cells, as it is done in most of the protocols, it provides already a shearing of the membrane to allow the enzymes to go in more easily. So if you don't do the flash freeze and use fresh extract, it's important to lyse your cells more to make sure that the enzymes can go in, because all the other steps are all gonna happen within the nucleus of your cells. So the next step is to cut your DNA. And for that, you have two ways. So you can either do it with restriction enzymes that are gonna cut at specific place on your genome, or you can use a microscopical nuclear enzymes that are gonna cut at random place of your genome where it is open. So there are two kits now that are commonly used for IC, the ARIMA kit, which I used and used the digestion enzymes, and the Dovetail kit, which used the myocroC. So the advantage of the myocroC, since it's cutting everywhere, the resolution you can reach is a lot higher, because with the enzymes, it's going to cut at specific places. So there are places where it's not going to cut. So if there's an interaction that starts exactly there, well, you're not going to have a cut site here, so you're not going to have a recombination there, so you're not going to see it. So you cannot reach a resolution that's as high with this than with the microC. However, since in this one, you know where you are cutting, you also know where your reads should be, so you can filter your reads more easily with an in situ ISC. So if you don't want a resolution that's incredibly high and that requires billions of reads, you're better off with the ARIMA kit and in situ ISC. Then you're going to need to do the fill-in and the ligation. So when you fill in, you're going to add the biotin mark that is going to tell you where your sites were cut, and then you ligate it. So if you don't have the biotin mark, when you're going to shear your DNA, you're going to have some segments that were sheared here, but also some that were sheared here, which is not a recombination read. So it's not a read that's going to provide you with info on the recombination of your chromatin. So it's a bad read. So if you don't do the biotin selection, only 30% of your reads are going to be recombination reads. So 70% of your sequencing is going to be useless. So even if your biotin costs a lot, you're going to save a lot in the sequencing if you do it. So I recommend that you do use a biotin selection, and the two kits I talked about do a biotin selection in their protocol. So now we cut the DNA, we marked it, we attach it, and now we extract the DNA, and then we sonicate it. You want a size of about 300 base pair when you sonicate, and after your size selection too. So it's important to optimize your sonication condition before by you using only some DNA, and your size selection to make sure that when you use your samples at the end, you're gonna have a fragment, well, you're going to have a series of fragments that are all around 300 base pair. And then we have to prepare the library. So we have our segment of DNA around 300 base pair with a biotin mark in the middle. So we select for the biotin with streptavidin beads. And the interaction between streptavidin and biotin is one of the strongest one in biology. So it's really hard to elude it. So when we do the library preparation, we do it straight on the beads. So if you're not using a kit specifically for ISC, you want to make sure that the library preparation kit you're using can work on DNA that is bound to beads. And the adapters you want to use some with barcodes so that all of them can be sequenced in the same flow as cells, which is going to reduce the cost of sequencing. Also, you want to make sure to optimize the PCR condition you're going to use before you do it, because if you use too many cycles of PCR, if there was a bias in the early cycles, after 20, 30, or 40 cycles, this bias is just going to drown all the other reads you're going to have. So you're going to have some sites with interaction that are incredibly high just because you amplified it more. So you usually want between 7 to 10 cycles, and it's good to optimize the conditions before. And then you're going to need to sequence your reads. So communicate with the sequencing facilities that is going to do the sequencing for you. You know how you need to ship your samples. Also, it's critical that you ask for paired end sequencing because you need to sequence both ends of your fragment. If you sequence only one, well, you're going to know that the fragment where this is going to map is attached to somewhere else on the genome, but you're not going to know what this somewhere else is. So you absolutely need the paired end. For the read length, the longer the read length, the easier it is to map. So if you want to look at interactions in regions of the genome that are highly repetitive, you're going to want a bigger read length to map it more easily. But if you just want to look at everywhere on the genome without any specifics, then a read length of about 50 base pair is going to be good enough to have a mapping that's going to be close to perfect. And then how many reads are you going to need for ISC? Well, it's recommended to have 2 biological replicates with around 300 million reads. So in total, you're going to have 600 million reads per condition. And with this, you can reach a resolution about 5 kb. So on a resolution that's a lot lower, you're going to need a lot more reads. So now, how do we analyze? So I know I talked a lot about the resolution of the ISC, the heat maps. Well, here I'm going to show you how to read those and how to make those. But first, well, we need to align the FASTQ files. So when you sequence, the sequencing facility is going to give you a text files that are the FASTQ files, which are basically just a list of the ATCGs found on your reads, and on one end and the other end. Then you're going to need to turn them into BAM files, which are going to be a file where these reads are aligned on the genome. So if in the FASTQ file you have ATC, GGC, well, then you align it. And on the BAM file, it's going to say, well, it's on chromosome 1 from 100 to 200. And the other end is on chromosome 1 from 500 to 6. So it's going to give you the coordinates. And for ISC, there are many pipelines to get the BAM files. So run IC or IC Pro or Juvisor, they're going to do the alignment for you. And they're also going to filter the reads for ISC because you want your reads to be one on one end of the genome and another one at another place on the genome. If both of your reads are too close together, well, it's a bad read. Just means that it's not a recombination, it's your sequence itself. So there are many things that can happen during the protocol to induce these biases, and the pipelines that analyze the IC are going to remove those. So it's only going to keep the good reads. And after you use the kit, the amount of valid reads you should have should be around 90% of your reads. And the rest, the 10%, are going to be either reads that ligate to themselves, that didn't ligate, but that were still selected with biotin. And then at the end of the pipeline, once your reads are aligned and you know one end is where and the other end is where, the software is going to create for you a contact matrix. So there's two format for it, either the NXN or the sparse. So the NXN is going to list all the contacts on each of the chromosomes. So for example, here on chromosome 1, between 0 to 10,000 kilobase pair, you had 2 interactions with 10 to the 20, but 0 from 20 to 30 and 0 from 20 to 40. And then in the sparse, it's the same as the NXN, however, it removes the 0 and it's just a list. So from 0 to 10, and then from 10 to 20, you had 2 interactions. From 10 to 20, and from 20 to 30, you had 4, and so on. So you're going to have one of them for each of the chromosome, and also for each of the combination of the chromosome. So here is going to be on chromosome 1, and here on chromosome 2, and it's going to list all the interactions. And it's with this that you're going to make the heat map. So how do you read a high-C heat map? Well, first, all the heat maps have a resolution that you're going to specify first. So you choose the resolution you want your IC heat map to be and your matrix to be. And this is going to be the length of your square. So each square here is going to be 10,000 kb by 10,000 kb. Then within those squares that we call bins, we have the digestion fragment. So these are where you know your enzyme was cutting, so where your reads should be. So if you use a micro C, you won't have this. It will just straight go to the reads. So then in each of the bins, well, you're gonna have one end of the reads that's mapping somewhere and the other end of the reads that's mapping somewhere else. So the interactions, it looks like this. So where you have some reads here, that goes in some reads there. So since you know you have an interaction between this one and this one, well, here is going to be a light pink because you have one in interactions. Here, since you have a lot between these two, it's going to be a darker shade of pink. So the darker it is, the more in interaction you have. And depending on the software you use to do your heat map, it's either going to be an absolute read or in normalized reads. However, the classical approach to bin the cells, you lose a lot of info here. Because if you look at the bin here, the contact between this and this is only between this part of the bin and this part of the bin. So here and here doesn't contact each other. However, with the 10 kb resolution, you still think that it does because everything here is pink. So if your resolution is lower, well, it's going to cut all your, if you use example 5,000 instead of 10, well, it's going to cut all your bins in half. Then you would see that only this section of the chromosome interact with this one. So here you would have other squares and only the square on top here would be pink. However, to increase the resolution, you need to increase the read counts. And to do that, you need to sequence more to be sure that in all bins, you have at least one read to have a coverage that's complete along your genome. But now there are other ways to calculate the interactions and to make the heat map to reach a resolution that's higher without having to have more reads to complete every bin by simply moving the bins around. So this is what the iFi is doing. It's a software made by Mathieu Blanchet's lab, and it moves the bins around to be able to detect specifically where the interactions are. So if we go back here, if instead of having the bin starting from 0 to 10 and then from 10 to 20, we had a bin that started from -5 to 5 and then from 5 to 15, we would see that the one from -5 to 5 interacts with the one from 35 to 45, but not the one from 5 to 10 and 45 to 55. So then by moving the bins around, it can know exactly what section of your bins is in interaction with which one. So without having to have more reads to complete your coverage, just by moving the bins around, it can increase the resolution very much. So I use that for the project I'm working on CBTCF. So as an example of how this works, here are my wild type cells. This is a segment of the chromosome 20 from 48 million to 48,750,000. And we said the wild type and the knockouts here, the interaction looks very similar. So we have here some dark bins here, some dark bins here. In the knockout, we see that it's a bit darker here than here. But by using the classical binning of approach, it's really hard to see if this is a clear change or not. However, with I5, they moved the bins around and they could pinpoint exactly what was the cause of this increase in intensity. And it's exactly this dot here that is not in the wild type. So how can this little dot be so important for a project? Well, it depends where it is. So it happens that this loop is located exactly between an interaction site where CTCF is lost at the promoter of snail, which was upregulated in our knockout, and an answer site where HK27 acetylation, which is a mark for active enhancer, was also gained. So basically this loop here shows that the loss site of CTCF leads to a loss of insulation that allows the enhancer to contact the promoter of snail to increase its transcription, which then increase its level in our cells, which drives its oncogenicity. So here, if I didn't add a higher resolution, I would have never found this specific site. And I couldn't have drawn a clear link between the loss of CBTCF and the increased expression of snail. And these dots are called peaks or loops, and there's a way to find them. So loops are going to be the darkest spot on your heat map, because when you do a loop, the boundaries of it is where it's going to be the closest together. So these are the darker points on your map. And the peak colors are used to identify those peaks. And now, the one that is commonly used is called Pikachu, like the Pokemon, and it uses a machine learning algorithm to detect these peaks. And what is nice with it is that you can train it to be specific for your model, because the peak colors are always going to vary depending on how many reads you have, the density of reads you will have. So one can be good for a set of reads, but really bad for another one. But with this, you can train it specifically for your sets of reads. And if you read the paper here and go in the sources, they give you all the models you're going to need to train it. So I won't go in the details of how you train an AI, because it would take really a big amount of time, but can give you a trick. So you don't want to undertrain or overtrain your Pikachu, because if you use a set to train it where all the peaks are not really clear, well, your Pikachu is going to be undertrained, so it's only going to be able to detect a peak if it's clearly there, if it's clearly a big peak. But if you overtrain it, so you train it with a set where all the peaks are super sharp, it's going to think that every dot that's a bit darker than the other one is a peak, so it's just going to call peaks everywhere. So just train your model, well, with many different ways, and see which one works the best for your peak, and you're going to have the best possible peak calling. So I don't have a figure to show for that yet, because I'm not done with the training of my Pikachu. However, for the next step to analyze, I do have some figures. So the next step you want to do when you analyze your data is using a TAD caller. So I've been talking about TAD since the beginning, where it's a region where the interaction within the TAD is stronger than outside of it. And now the TAD caller can also call TADs within TADs. So here it detects that the interaction within this is bigger than outside of it, but the interaction here is even bigger than the interaction there. So it can tell sub-TADs. And it not only calls where your TADs are, but also the boundaries. So if you remember, my project focuses on CTCF found on the boundaries of TADs. And I wanted to draw a clear link between the changes in CTCF and the changes in gene expression. So I wanted to see if the changes in boundaries, so how the TADs are changing, if it's associated with my changes in gene expression, and it does. So when I look at the altered boundaries in my knockouts, I can see that the genes that are enriched around the gain boundaries are significantly enriched for the PI3K pathway, which was up in my knockouts and driving the oncogenicity of it. And the other pathways I see in the gain or the lost boundaries are also found in my RNA-seq. So there is a clear link between the change in boundaries and the change in gene expression. So to give a more specific example of that, if we look at NTRK2, which was the top hit on our heat map for PI3K pathway genes and is an oncogene that is known to activate PI3K pathway, we see that in the wild type, the promoter of NTRK2 is found within a subTAD here that isolates it from an answer that is found here that is in another domain. In our knockout, where the binding of CTCF is altered here and here, we see that the isolation that happened here is lost. So now it can contact the enhancer here, which is then in contact with the promoter and increase its expression. And we can see this gain of interaction as the smear here. Because when an enhancer is going to look for a promoter, it's going to scan the sequence before it until it finds the promoter. And we can see the scanning here because as the IC is done with millions of cells, in some of the cells, the enhancer was midway through its scanning, so here, and in other cells was straight on the promoter here. So these smears are a representation of this enhancer scanning and of the dynamic regulation and upregulation of this gene in the knockout, allowed by the loss of insulation here. And also that's going to be it for my seminar. So basically, with the IC, I'm able to link the loss sites of CTCF to the changes in gene expression by showing that the loss sites induce change in conformation that allow the enhancers that were once insulated by CTCF now to contact the genes. And then these changes in gene expression promote the oncogenicity of our cells, which promote cancer progression. And that's it. So thanks.
Thank you so much, Benjamin. I think that was a wonderful presentation, and your narrative was really easy to follow, and I'm impressed by how much you were able to cover in such a short presentation. So, I mean, I, for one, wasn't actually aware of the enhancer scanning method, so that's one for my notes. So before we go ahead with your questions, I just wanted to remind everyone who came late to the presentation that this meeting was recorded. So if you missed the introduction or any other part of the presentation, you should be able to watch it on emgss.org/TSS. So that's for Technical Seminar Series. I'll hopefully be able to upload it later today. EMGSS, GAP, and GAPTS will be hosting our technical seminar series in December as well, but the exact date hasn't yet been determined, so you should be able to see an update about that on our website, as well as in listservs to come. So I'll open the floor for questions if anyone has any. You can put them in the chat or raise your hand and just open your microphone.
I have a question.
Yep.
So, well, first, thank you, Benjamin. It was really nice, your presentation. I've never heard before about this technique, so I'm wondering if you can assess for other things that are not promoters. So, for example, I'm thinking I work with retrotransposons, so I'm wondering if I could be using that technique to identify if there are like retrotransposition happening. So it will be like the proteins for each retrotransposon binding to their own mRNA and the genome of the cell.
Yeah, so it's really it's not just used for the promoter and an answer, but it's used for the structure of the DNA. So it really needs to be in contact with one of another. So I'm not an expert in retrotransposons, but do they change the structure of the chromatin?
No, they bind. They cut the DNA.
Yeah, because if it's just binding, then a chip would be better. Because with a chip seq, you can see where either a protein or is binding to the DNA and where it is binding. The IC, it's really it's telling you where one part of the genome is physically in contact with another part.
Yeah, I was thinking more for identifying where it is inserting.
Oh. Yeah, I don't know if it would work for that. I never thought about it.
Okay, well, thank you.
So Benjamin, I can just read out a question from the chat. So thanks for the great presentation. And Sophie says, with the in situ method, do you get enough coverage in regions that are more compacted or less accessible for the enzymes used in the earliest steps of the protocol?
Yes, so your coverage is going to be worse in those because it's going to be harder for the enzymes to cut there. So if you're in a terrested specifically in the dose, you're going to need to have a lot more reads. So you're going to need to sequence more, but you will have enough in your DNA to map it in the region there, and it's going to cut there. But the regions that are open and more easily cut are going to drown this signal. But if your sequencing is deep enough, you're still going to have enough reads to see them. Yeah, I cannot see the chat, so is there any other questions?
No other questions. I guess on that note, I'm curious if you, I haven't really formulated this question. If you're looking to understand interactions between maybe more compact and less compact, I'm wondering how swayed that might potentially be by say the over-sequencing of less compact regions.
Yeah, that is a good question. Yeah, I'm not sure how to answer that because it will cause bias, but I don't know if it would be big enough that it would really do a major change.
Yeah, I wasn't sure about that exactly.
Can I ask a question?
Oh yeah, sure.
Hi, Benjamin. So I'm Mike Witcher, Benjamin's supervisor. So actually, I think the last question was relevant and actually may highlight a bias of the technique itself. I'm wondering, when you digest the genome with specific restriction enzymes, do they cut equally throughout the genome at euchromatic versus heterochromatic regions, at open versus closed regions? If they do, then you would expect for the technique not to have a bias maybe for open versus closed. But if they don't cut equally with equal efficiency at open versus closed regions, that may introduce a euchromatic versus heterochromatic bias. So can you comment on that?
Yeah, there is one. So the regions that are more open are always going to have a coverage that's deeper because you're going to have more reads there. So when you analyze it, the software that analyzes it are going to have the regions that are usually more closed and open to try to compensate this bias, but there is a bias still because it's going to vary from cell line to cell line. So you're always going to have a coverage that's better in a region that's open than a region that is closed.
Okay.
But you're still going to have reads in the regions that are closed. Just the intensity of your heat map at this part is always going to be weaker. So to make sure that you detect all the interactions there, you need a sequencing that's a lot deeper if you really want to specifically look at the smaller interactions within the closed states of chromatin.
Okay, so that's clear. Thank you.
Yeah, any other?
I guess I have one more question. You showed that CTCF knockdowns still do have some partitioning in their chromatin structure. I'm curious how that structure might still arise. Is that just localization within the nucleus, or are there other proteins involved?
So you mean a knockout that will wipe out all the CTCF?
Oh, I guess what you were showing was knockdowns where there's still a little bit to partition the.
Yeah, so in the knockouts that I have, it's hemizygous. Only one allele of CTCF is knocked down to a reduction of the protein level of about 60%. But in every cells, there's more CTCF than what they need. So you don't have a loss of 60% of the binding of your CTCF. It's more about 5,000 sites out of about 35,000. So the structure overall, if you just look at the chromosome and the big TADs, it's not changing much. But if you look at the sub-TADs, that is where the change are happening more. Because the binding there is weaker for CTCF, so the stronger sites that bind strongly to a CTCF are still going to keep the binding, but the weaker sites, they won't be able to bind the lower amount of CTCF that's in the cell. If you do a knockout completely of CTCF, then you still have the coevasin that is going to form the TADs. It's just not going to be stopped by CTCF. So your TADs are all going to be merged from one of another until the coevasin contacts another coevasin or RNA pol 2 or another structure on it. So without the CTCF, you still have some TADs. They're just going to be merged together.
Yeah, I appreciate that. Thank you. So unless anyone else has any questions, I think that's it for today. I really appreciate it. Thank you again, Benjamin. Oh, sorry,
Thanks.
there was one more question. Can I ask just where the recording will be uploaded? Oh, so yeah, again, for anyone who missed the beginning of the presentation or have friends who wanted to see it, you can always check it out at emgss.org/TSS. So TSS for Technical Seminar Series and EMGSS for the Experimental Medicine Graduate Student Society. So yeah, again, thank you, Benjamin. If anyone has questions specifically related to the methods used, you should contact Benjamin. If you have questions related to the Technical Seminar Series, you can contact me. My information is also on emgss.org. So, well, thank you very much. Bye, everyone.
Thanks.